discuss this topic in unit 9.1. So in paper 1 anthropology, that is in paper 1 anthropology, we will be discussing this topic in unit 9 in physical anthropology. So we have already finished pedigree analysis long back. So we also discussed a little bit about what are twins. We are yet to discuss twin studies, co-twin method. So we will discuss that chapter first now. Okay, now unit 9.1, twin studies. Twin studies. So in our previous lecture itself, we discussed the two different kinds of twins, right? That is the monozygotic twin and the dizygotic twins. So when two different eggs or ovum are fertilized by two different sperms, we are getting the resultant as dizygotic twins. The resulting twins are dizygotic twins, right? And when a single egg or the ovum is fertilized by a single sperm, and then after formation of the zygote or the embryo, then this embryo divides into two, giving rise to two different kids, then we call it as monozygotic twins. Isn't it? We discussed all of this in our previous lecture itself. What is monozygotic twin, what is dizygotic twin, and what is concordance and what is discordance. So now we shall move on to the twin method and co-twin method. In the syllabus it says twin study and co-twin method. Twin studies and co-twin method. So as I already told you, we have two kinds of twins, that is the monozygotic twin which share around 100% of their DNA. So they have the similarity in the genes because they are originating from a single embryo or from a single zygote. Then we have the dizygotic twins. Okay, the dizygotic twins hardly share around 50% of the genes. So now the twin method is based on the fact that biological peculiarity of monozygotic twin originates from the division of the zygote. Okay, so twin studies have typically been used to determine whether genetic factors play a role in etiology of the disease. So twin study is used to identify whether it is the genetic factors which are responsible for a disease in the twin. Okay, so here both in twin studies and co-twin method, what we are trying to understand is to what extent genetics determines the disease in a twin and to what extent environmental factors influence the twin. Okay, so twin study are used to determine whether genetic factors play a role in the etiology of the disease. So why we are studying this is because we need to understand in the case of monozygotic twin, they share around 100% of the genes. So if suppose they are getting the disease, we can attribute it to their genetic makeup. Whereas in a dizygotic twin, if they are getting the disease, there might be the possibility of genetic influences because they share around 50% of the DNA, and there might be environmental factors also. So we want to understand whether it is the environmental factors which are responsible for producing the disease or is it the genetic factors which are responsible for producing the disease. So that is what we are trying to learn by studying the twin study and the co-twin method. So twin studies are used to determine whether genetic factors play a major role. So is it the genetic factors which play a major role in the etiology of the disease? So etiology means history of the disease, right? So if a twin is getting a particular disease, we want to understand to what role, to what extent the genetics of them is playing the role in them getting the disease. So I already told you in case of monozygotic twins, or in case of both twins also, but most commonly you can find that in case of monozygotic twin, if one child gets a disease, within some time, you cannot say disease exactly, say a common cold or a fever or any other disease, so immediately the other child will also get. So in case of many other diseases also, you can find if one individual among the twin is getting the disease, the other one also will definitely get the disease. So in these cases, we can attribute it to the genetics because they share 100% of the genetic makeup. Whereas in dizygotic twins, if say one person, only one individual among the twin gets a disease and the other one doesn't get, then we can say probably it is due to the environmental factors. Or if both of them get the disease, then we can say probably it is because of the genetic factors, because both of them share at least 50% of the genes, right? So what we are trying to understand is, monozygotic twins share 100% of their genetic material, whereas dizygotic twins share on an average 50% of the genetic material. So if the set of twins are being studied, the monozygotic twins are found to be concordant, isn't it? So what do we mean by concordance? If a particular trait or a character is expressed by both the twins, or it is not expressed by both the twins, then we call the twins to be concordant. If a particular trait or a character is expressed by one individual of the twin and it is not expressed by the other individual belonging to the pair of twins, then we call them as discordant. I even gave you the example of eye color. Say if both the twins are having brown eyes or black eyes or same colored eyes, then we can say they are concordant. Whereas one individual of the twins is having blue eyes and the other one is having brown eyes, then you can say they are discordant. So just I am giving you the example of eyes for you to understand it properly. Just understand that concordance means if a particular trait or a character is expressed by both the members belonging to the pair of twins, then we call them as concordant. So in concordant twins, so what they are saying is, if monozygotic twins are found to be concordant, that is they have the same disease, they have greater frequency than dizygotic twins. Okay, so because monozygotic twins share 100% of the 100% of the DNA or 100% of the genes, you can find that monozygotic twins are having a higher frequency of concordance and in comparison to the dizygotic twins. Got it? So the percentage of concordance among twins is high among the monozygotic twins in comparison to the dizygotic twins. Okay. So the classical twin method examines as to what extent genetics and environmental factors contribute to the variation in a trait. So here we are trying to understand to what extent genetics contribute and to what extent environment contributes in the variation of a trait among the twins. So to what extent the genetics is responsible for a particular trait in both the twins, or to what extent the environment is playing a role in forming a particular trait or character among the twins. This is what the co-twin method is trying to examine. So according to the co-twin method, the premise or the basic of this is that the monozygotic twins not only share all their genes, but also they are bringing up an early environment. So I told you in the case of monozygotic twins, because they are formed as a result of the cleavage, or no, you cannot say cleavage, division. Okay, the zygote is formed. After the formation of zygote, it will divide into two and it will result in two individuals, that is the twins. So the premise of twin method is that monozygotic twins, as I already told you, they share around 100% of their DNA. In addition to that, they also the early environment. So early environment here means that is the environment inside the fetus, isn't it? So they have a common sac which is holding them together. So it is the environment also which is responsible for that. Okay, so the classical twin method examines to what extent the genetic and environmental factors contribute to the variation of a trait, and it says that monozygotic twins not just share their DNA or the genetic material, but they also have a similarity in the upbringing and the early environment. Okay. So, however, you can see that in the case of dizygotic twins, they hardly share around 50% of their DNA, and in addition to that, they do not have a similarity in the early environment, because from the very beginning there are two eggs which are getting fertilized by two different sperms, and after the zygote formation they have separate sacs, isn't it? So even the early environment is not similar for them. So here you can see that the phenotypic dissimilarity between the monozygotic twins is assumed due to the environmental differences between the twins. So if there is any changes or a dissimilarities in the phenotype, okay, what do we mean by phenotype? Phenotype means external expression, right? Genotype means genetic expression. Phenotype means external appearance, which we can see, be it in the shape of the nose, shape of the ears, the texture of the hair, color of the hair, eye color, all these are phenotypic expressions, isn't it? So the phenotypic dissimilarity, sometimes you can find that even among monozygotic twins, there will be certain dissimilarity. You can identify, okay, even though they look similar, they are not exactly 100% similar. To certain extent, at least after few days of mingling with them, you can identify who is who, right? Who is suppose there are two names, who is this and who is A, who is B, you can identify. So they say that it is because of the dissimilarity in the environmental differences. Okay, so monozygotic twins dissimilarity is assumed due to non-shared environmental differences between the twins, whereas dissimilarity between the dizygotic twins is assumed due to both genetic and non-shared environmental differences. So in case of, suppose there is a dissimilarity among the monozygotic twins, they attribute that to the dissimilarity in the environmental factors. Okay, so when there is a dissimilarity in the monozygotic twins, they say it is due to the environmental factors, dissimilarity in the environmental factors, whereas when there is a dissimilarity between the dizygotic twins, they attribute that to both genetic as well as environmental factors, because genetically also it is true, isn't it? In dizygotic twins, they hardly share around 50% of the genes. So any trait which is dissimilar among them might be due to genetics also, or it might be due to environmental differences also. Okay, so if the monozygotic twins are similar for a trait than dizygotic twins, then a genetic contribution to the trait can be inferred. So when there are monozygotic twins who are sharing most of the traits, they have a similarity in the traits, then we attribute that to the genetics, because they are sharing around 100% of their genes, right? Under monozygotic condition, the twins are sharing 100% of the gene. So when there is a very high similarity in the characters or traits, then we can say it is because of the genetics. Whereas here, if there is a similarity in the dizygotic twins, if there is a similarity in the traits between both the twins, then we can say monozygotic twins. Okay, did you understand this? So in monozygotic twins, they share 100% of their DNA, and dizygotic twins, they share only 50% of their DNA. So when there is a similarity in the monozygotic twins, we say it is because of the genetics. When there is a dissimilarity, we say it might be because of the environmental factors. And in dizygotic twins, when there is a similarity, okay, but that is probably because of the 50% of the genes which they share, and when there is dissimilarity, we say it is both due to the environmental factors as well as the genetic factors, because still the remaining 50% of the genes are not shared by them and they are different. This is what we study in twin study and co-twin method. That is, basically we are trying to understand whether environment plays an important role or genetics plays an important role in the etiology of a disease among the monozygotic and the dizygotic twins. Okay, so various experiments have been used on monozygotic and dizygotic twins, in order to separate the hereditary and environmental components of a character. So using the co-twin method, we conducted various experiments on both monozygotic and dizygotic twins in order to separate, that is, to what extent hereditary is responsible, okay, for the formation of a character in the twins, and to what extent environment is playing a role in the formation of a character in that particular twins. This is what we are doing in co-twin method. Now let's move on to foster child. Let's move on to foster child. Foster child. What does the word foster mean? Actually, now it is mostly preferred to use adopted instead of foster, because foster is somewhat kind of hurting for many people, right? It's not exactly derogatory, but to a certain extent it hurts people. So actually what foster meant was, in Western societies, when a child is orphaned, so he will be left under the custody of foster parents. So the foster parents will take care of the child for, it is not a permanent adoption where the child is completely given to the parents and they raise them for a lifetime, and it's not like that. So there have been many stories of people in Western societies where they have been raised under several foster parents. So for 1, 2, 3 years they'll be under the guidance of one set of parents, then they will move on to another set of parents. So in many countries, even the government provides support to the parents, that is monetary support to the parent to raise the kids. Okay, so depending on the age of the child, the government will produce some kind of stipend or some kind of monetary support for them to raise the child. Then after 3 to 4 years or so, they will be moving on to another foster parent. So this is what. So it is similar to adoption, but it is not a long term, isn't it? So I'm sure all of you would have watched this movie. I don't know if I should tell this in class. Okay, so I'm sure you would have forget that movie. Let's not speak about that movie here. At least you would have read this novel, They Both Die at the End. By Adam Silvera. So here he gave, in that novel they'll give you about description, so there are 1 or 2 chapters about foster home. So how children grow up over there and all these things. Then another movie also, the hero is another famous English movie. The hero is, he would have been taken out from the foster home, and we have novel also on that. Okay, let's not go into that. Fine. So foster child. Foster child means adopted child. Under that, we have 2 studies. One is the Chicago studies and the another one is Minnesota graded home study. First we shall discuss Chicago studies. Chicago studies. So here in this study, groups of children were chosen randomly and they were placed into different homes. So one group of children randomly they were chosen and they were placed into 3 different kinds of home. First one is good homes, second one is average home, and third one is the poor home. Okay, good home, average home and poor home. So now you might think, what is good home, what is average home, what is poor home? You can assume like a good home means which has a luxurious kind of living with all the modern gadgets, modern education, very good environment for studies and all that. Average means you can understand the middle class life of Indians. That has as an average home. And then poor means you can think it's like the BPL, below poverty line, where they do not have access to even basic things, forget computer or education or going to schools, very, very minimal things they will be living out of. So this is what we can understand by good homes, average home and poor homes. So randomly a group of people were chosen and they were placed into these three homes, that is good homes, average home and poor homes. And then after a certain point of time, so they gave them some time to stay over there, some time means few months or few years also, and then afterwards they were tested for intelligence. So they were administered some tests like for testing their intelligence, and it is said that if the environment of the adoptive homes influence the children, then they will exhibit different scores in IQ test. Say we have this, always have this confusion, right? Some of them say intelligence is based on genetics. That is why we say Jews. You can see most of the top think tanks in USA are Jews. Even though Israel is such a small country and the population of Jews is very, very tiny in the world, you can find that throughout the world, throughout the globe, wherever the top think tanks and topmost positions, wherever, it is mostly occupied by the Jews, because it is said that by nature they are highly intelligent people, isn't it? We also believe in this. So when somebody performs very well in exams, they read. It is true also. See, some people you can find that they will slog and read throughout the year from morning till night. They'll prepare for 10 hours, 12 hours, yet they will not be able to clear prelims. Whereas some people just, after the prelims application comes, they will apply for it, and then some 2-3 months they will sit, they will not take too much of stress, just they will read everything, and finally they manage to clear prelims, and finally they get into the list also. Isn't it? So then we'll say, what is it? Is it because of the genetics, right? So we always have this belief. Sometimes we say it is because of the environment. We say, okay, they have a very good environment. See, people in rich homes, like you can consider the example of our Ambani's children. They have the best of everything, right? There's no need for them from a very young age to stress about or take stress about anything. They get the best of, right from, if they want to study in some Ivy League college or school, they can get it. Right? They can get the best technologies, everything is at their fingertips. So obviously their level of exposure and the way how their intelligence is will be much better than compared to the other people, right? So we have this always this idea that it is the genetics which influences our intelligence, and sometimes you can find children belonging to parents who are very highly educated. So say there's a couple who was from both from IITs or IIMs, or both are civil servants, civil service IAS or IPS couple. So you can find that automatically their children also will be very much intelligent, right? And we also watched that movie, what is that, Chickchore, isn't it? So it is not easy to be a child of very successful or parents who have achieved a lot in their life, because the society will put pressure on you also, because you have to live up to their expectations. You also should be highly achieving just like your parents, isn't it? Okay, let's get back to our Chicago studies. So after a time lapse, when they tried to test these three groups of children, they found that if there is the influence of environment on the intelligence, then they should score better, isn't it? Suppose it is the environment which is playing an important role in the intelligence character, then the children placed in good homes should have very high IQ scores, and the children placed in average homes should have less, and then the children placed in poor homes should have very less intelligence. This is what we expect if we say that it is the environment which plays a role in the formation of this character called as influence. So in the Chicago study, it reveals that in good homes, 45 adopted children scored 112 points. 45 adopted children scored 112 points, and then in average homes the children scored 39 points. Okay, sorry, 105 points. 39 children scored 105 points. Okay, and then in the poor homes, 27 adopted children scored an average of 96 points in the intelligence test. So what does this reveal? It reveals that yes, environment definitely plays an important role in the intelligence of a child, because in the good homes you can find that they have a very high score, that is 112. In average homes 105, in poor homes 96. Okay, so it says that environment definitely has a noticeable effect on the development of intelligence. But again, we must also keep mind that there is possibility of human error also in these experiments. Say when they say we are taking a random group of children and we are placing them in good homes or average homes or poor homes, you don't know who those random children are, or whether they are actually placing those children, without bias into the good, average and poor homes. So we have to keep in consideration that also. So this experiment is not completely free of bias and error. So there might have been a possibility that they would have taken very children from very good families only and placed them in good homes. Okay, whereas poor children, children who are very poor and low in IQ, they would have placed them in poor homes. So this is also a possibility, but overall there is certain effect, or there is a certain noticeable effect of environment on the development of intelligence, that we can see even in our society. Forget Chicago studies, even in our very own society you can see very rarely, randomly you will find someone from a very low economic background cracking UPSC exam, or getting into IITs, or getting into IIMs, or becoming a scientist or something. But overall that is not the case, isn't it? That happens once in a while. So out of some millions of people, one of them will be like that. So even though they were born in a family full of poverty, they did not have any proper means of education, proper schooling or books and facilities for overall development, still they turn out to be achievers. But you can see most of the times it is the children who are living in these good homes, in the upper middle class or in the high class society, in the rich class, in the affluent class, they have very good intelligence because they have very good access to good educational facilities. They have motivating parents who can guide them, who can teach them whatever they don't know, isn't it? So all these also play an important role. So environment definitely influences the development of intelligence. Fine. Next, moving on to the Minnesota graded home study. Minnesota graded home study. Minnesota graded home study. So here, this study is to determine the role of heredity in the development of IQ. So earlier in Chicago studies, we tried to understand what is the influence of environment in the development of IQ. Here in Minnesota graded home study, we are trying to understand what is the influence of hereditary in the formation of the trait called as intelligence. So to what extent genetics plays a role in intelligence. Okay, so here what we did was we took children from managerial class. Okay, so we took children from managerial class and we placed them in labor class homes. Okay, then we took children from labor class and we placed them in managerial class homes. Okay, so we took children belonging to managerial class and placed them in labor class homes, and we did the vice versa. So now actually the children of managerial class had more IQ even after being placed in labor class homes. This explains the importance of heredity in the development of IQ. So, am I teaching very fast? Are you all following? Are we together? See, honestly speaking, these are very boring topics. Okay, this entire twin study, co-twin method, biochemical methods, immunological methods. Okay, it's just, and it's very factual also. You just need to read it once. I could have just given you the material and asked you to go through, but still I thought it will be better to teach, and if I speak very slowly you might fall asleep, that too after a good lunch. Did you all have lunch? That is why I'm going a bit fast. Not fast, okay, pace, I guess. What do you say? Okay, back to Minnesota graded home study. So in the study, we are trying to understand the influence of genetics, or how, to what extent genetics influences our intelligence. So what they did was they took children from managerial class and they placed them in labor class homes. Then they took children from labor class homes and placed them in managerial class homes. So after a certain time, so say after few months or few years, they will do an intelligence test and they will try to understand what is their intelligence level. So here they found that the children belonging to managerial class, okay, the children belonging to the managerial class, even though they were placed in labor class homes, they had a much better intelligence. That is, they had better IQ in comparison to the children who belonged to labor class and they were placed in managerial class homes. Okay, so originally what we think, if it is only the environment which is influencing the formation of a trait, if it is only environment which is influencing our intelligence, then what should happen? These children, the labor class children, when they were placed in managerial class homes, they should have high IQ, isn't it? And these children who belong to managerial class, when they were placed in this labor class homes, their IQ should have gone down. So, but actually that did not happen. So even though the managerial class children were placed in labor class homes for some time and afterwards they were tested, we found that these children, that is the children originally belonging to the managerial class, they had a higher IQ in comparison to these children, that is the labor class children. Okay, so what we understand from this, we understand that genetics also plays a major role in the formation of intelligence. So in Chicago studies, we said that environment plays an important role. Okay, environment plays a role in the formation of intelligence, and here we say that it is the genetics which plays a role in the development of intelligence in an individual. So now confidently we can say it is a, you can say that both genetics as well as environment help us in intelligence, isn't it? Okay. Next we shall move on to, these are all very simple topics. I hope you all are following, right? What is twins? What is monozygotic twin? What is dizygotic twin? What is twin studies and co-twin method? What is it that we are trying to understand over there? We are trying to understand whether genetic plays a role or environment plays a role in the etiology of the disease formation in the twins. So we are trying to understand that using the twin and co-twin method. Then further we discuss the foster child. What is meaning of foster child? That is adopted child, right? So using Chicago studies, we try to understand the, to what extent environment plays a role in intelligence. And then in Minnesota graded home study, we try to understand to what extent genetics plays a role in intelligence. It's as simple as that. Fine. Shall we proceed? Now moving over to genetic testing methodologies. Under that we shall be discussing the cytogenetic method. Yeah, cytogenetics. What do we mean by cytogenetics? Cytogenetics or cytogenetic method Okay. Moving on to cytogenetic method. So cytogenetics is the field of study which deals with the chromosomes and related abnormalities. We discussed a lot of chromosomal aberrations, isn't it? Down syndrome, Edwards syndrome, Patau syndrome, super male, super female, all this. So cytogenetics is the study of the chromosomes and the abnormalities related to chromosomes. This is what we call it as cytogenetics. So chromosomal analysis is also known as karyotyping. I showed you how karyotyping is done, right? So how a karyotype chart looks like. In a previous class, when we discussed chromosomal aberrations, I showed you how a karyotype looks like. So there will be a sheet like this, and then here we will arrange the homologous chromosomes 1, 2, 3, right? So 1, 2, 3, 4 like this we will arrange. We will have a normal karyotype chart and we'll have the patient's karyotype chart, and we will compare both of them and try to identify what are the abnormalities that is present, and based on that we identify which disease they are suffering or which abnormality they are suffering from, isn't it? So cytogenetics involves the examination of the chromosomes to identify the structural abnormalities. Okay, so what happened in case of 5p minus, that is Cry-Ducat syndrome? One chromosome will be normal, and in the other chromosome, in one arm, a part of the chromosome will be missing. That is this part over here which was supposed to be there, that will be missing. That is called as 5p minus, isn't it? 5p minus, it is a structural aberration. So cytogenetics involves the examination of the chromosomes to identify the structural abnormalities. So how do we exactly do this? I will show you some pictures. That's not clear. I have already shared these pictures on WhatsApp. So in case the picture is not clear here, you can check your mobile. You will have the pictures on the WhatsApp group. So this is how karyotyping is done. Okay, so this is how karyotyping is done. Okay, so they will take out some cells, they will culture it, and they will do the karyotyping, and they will observe the results. This is how a karyotype chart will look like. Fine. Then see, again, these are various steps by which karyotyping is done. So sample is collected, the cells are cultured in vitro. What do you mean by in vitro? In vitro means inside the lab, right? So sample is collected, cells are cultured, and preparing the metaphase chromosomes. This we'll be discussing further later in this chapter itself. So how we can arrest the cells which are dividing? We need to arrest the cells at the metaphase plate. Okay, so we know that actively dividing cells, the chromosomes become condensed and clearly visible, and especially during the metaphase it is very clearly visible. So when you are trying to study, it does not take years and years for it to move from metaphase to next phase. Okay, we have prophase, metaphase, anaphase, telophase. So immediately after metaphase, it will move into anaphase. So if we want to study that, it will not wait for us to sit, to sit and analyze it properly. So we need to arrest it. That we'll be discussing later in this chapter itself. So we will be preparing the metaphase chromosomes, then some treatments, and then we'll make the stains. We'll stain it using staining agents. Then we will observe it under the microscope. Fine. So this is how the karyotyping is done. So this is another picture of karyotyping. Okay, so again here you can see how the sample is taken. It is cultured in the lab. See, again colchicine is added. So what is colchicine? Colchicine is a component which arrests the cell division. Okay, so we'll speak about that later. And the cells are fixed on the slide, and then we observe it under the microscope, and finally we make the karyotype chart. Got it? This is fish. What do we mean by fish? This is fish, not the other fish which we see in aquarium, or not the fish which you put on your plate and consume. Okay, this fish is fluorescent in situ hybridization. Okay, fluorescent in situ hybridization. So this is a process that paints the chromosomes or portions of the chromosome with fluorescent molecules to identify the chromosomal abnormalities. So we have chromosomal abnormalities when there's an insertion or a deletion or translocation. So any of these has happened, and when there's an abnormality in the chromosome, how do we identify that? So we can use this technique called as FISH, which is fluorescent in situ hybridization. Okay, so here what happens is the portions of the chromosome which have the defect, say be it a deletion or insertion or translocation, whatever, there is a defect in that particular part of the chromosome, only those part will fluoresce. Okay, so we will use fluorescent molecules. So those fluorescent molecules will, only those regions which have the defect, only that part of the chromosomes will start fluorescing. You understand what's the meaning of fluorescent, right? We have even fluorescent yellow, green, blue. We even call it as neon colors, right? So you know what is fluorescent. So only those regions, okay, you can by looking at that, you can identify that there's a defect over here. Only those regions will fluoresce. This is the FISH, or the fluorescent in situ hybridization technique. So it is used to identify specific chromosomal deletions. Okay. Next, moving on to biochemical method. Biochemicals, the chemicals secreted in our body, okay, the chemicals produced in our body are called as biochemicals, isn't it? Chemicals which are produced in our body are called as biochemicals. So these biochemicals may be enzymes, hormones, proteins, isn't it? Most of all enzymes are again proteins only. So they might, these chemicals which are produced in a body, such that the enzymes, hormones, proteins, okay, and DNA, RNA, all these are biochemicals. Okay, the study of this is called as biochemical method. It's as simple as that, isn't it? What is biochemical method? The study of the chemicals produced in a body. The chemicals can be enzymes or hormones or DNA or protein or any such components. So studying that is all about biochemistry or biochemical method. So we have various tests, okay, various techniques and various tests which we utilize to study the biochemicals produced in a body. So they are liquid chromatography, okay, which we call it as HPCL, HPLC. So HPLC means high performance liquid chromatography, gas chromatography, mass spectrometry, tandem mass spectrometry, and so on. So we also have special techniques such as chromatography, okay, electrophoresis, okay. So chromatography is an important technique which enables the separation, identification and purification of the components of a mixture for qualitative and quantitative analysis. So if you're any of you are from life, your background in degree was BSc or MSc, definitely you would have done these chromatography experiments. The first and foremost simple experiment which we used to do is paper chromatography. So what we would do is we used to take the chromatography paper, okay, we used to make a mark over here and we used to make different dots. So we used to do all this paper chromatography, electrophoresis, all this as part of our practicals in both BSc and MSc. So a paper is taken, a chromatography paper. This is how the paper chromatography is done. Okay, so we'll mark a line over here, and here we'll put different points. So different components are loaded over here, and after that we place this paper in a beaker. Okay, we place it in a beaker containing a medium like liquid. Okay, so then what will happen? These samples which we have loaded here will start moving like this. So we leave it for say 5 hours. So again depends on what your aim of the experiment is. Okay, so mostly the chromatography is a technique which is used for separation, identification and purification of the components and to study their qualitative and quantitative analysis. and after some time they will move in different places. So then we will take out this paper and we will measure the distance, that is from here. Say when we take out the paper, chromatography paper, it will be like this. So this will be the starting point where you have loaded your samples. Say this sample has moved till here, the other one has moved till here, the other one has moved till here. So we will measure this distance. So what is the distance which they have moved, and we have formulas. RF is equal to something. So we will put these measurements over here and we will arrive at the values. This is what we used to do in paper chromatography. So I'll show you pictures of paper chromatography. This is fish. This is how paper chromatography is done. So I told you already. See, this is a chromatography paper. This is the buffer solution. Okay. So we load the sample. See here we have marked already, the places have been marked. So we will load our samples on this paper, and after 5 minutes they would have moved so much. After 10 minutes they move further, and see RF. So then we will measure. Then we will take out this chromatography paper for after completely. There we have different timings for different experiments. So after so many hours, when completely it has moved, we will remove that chromatographic paper out of the buffer solution. We will allow it to dry. After drying, we will measure a distance. That is, it started from here, and after 5 minutes how much it has moved, after 10 minutes how much it has moved. So totally after what is the entire time duration which we kept it inside the buffer solution. So how much it has moved by then. So all these measurements we will take, okay, how many centimeters, and the same with the other sample also. Then we will put it in this formula and we will calculate it. Okay, so here you can see how this is the one which is the chromatography paper which we had placed after loading the samples. We have placed in the buffer solution. So from here it has moved till here. So second sample has moved till here, third one has moved till here, fourth one has moved till here. Can you see? So here we will put a mark like this and we will measure this distance. So moving on to electrophoresis. So electrophoresis, we do it for electrophoresis is again experiment which we used to carry out many times. It's a part of the experiment for many practicals. So we used to do it almost every year in our BSc and MSc. So what here we do is we prepare an agarose gel. What is agarose? Where is agarose derived from? Do you know what is agarose? What it is used for? See, agarose is extracted from weeds, seaweeds. Okay, and then it will be in a powder form. Then using the methods in lab, we will make a solution kind of. Okay, so that we will pour it into a slab, and after leaving it for some time it will solidify, like similar to our jelly bellies. We make jellies at home, isn't it? So we get this ready-made packets in the grocery stores. We buy that and they would have mentioned how much of water you should add, and you mix both of them, you heat it I think we have to heat it. Okay, so we have to heat it, and after heating we have to pour it into the molds, and slowly as it cools, after cooling, when you remove it, it is in a jelly form. It has acquired that shape. So similarly, we make the agarose gel for this electrophoresis. So agarose gel electrophoresis is a technique used to separate DNA, RNA and protein molecules based on their size and electrical charge. So what is the charge of DNA and RNA? Tell me. So for what do we use agarose gel electrophoresis? Agarose gel electrophoresis is used to separate DNA, RNA and protein molecules based on their size and charge. So both DNA and RNA are negatively charged. Okay, so what is the charge of DNA? Both DNA and RNA are negatively charged. So we used to take a agarose powder and we used to make a solution of agarose, okay, in a conical flask. Then we used to pour it inside a slab, that is the agarose gel unit, okay. Then after pouring it, after it becomes a proper slab, we will make holes in that. Say this is the agarose. I'm becoming very bad at diagrams, so better I show you the diagram which I have instead of writing everything and making, taking more time. So I'll just show you how the agarose gel electrophoresis unit looks like. Here. So this is how, see, this is how the agarose gel is made for electrophoresis. See, very simple. You don't break your head thinking how all these things are. Very simple terms, understand. Agarose gel electrophoresis is a technique through which we use to separate DNA, RNA and protein molecules based on their size and charge. So DNA and RNA are negatively charged. So what happens is we will take the agarose powder and we will mix it in water or buffer solution, when I forgot long time back. Okay, so don't worry about exactly how it is done. So you have to make the agarose mixture and you have to microwave it. Then after it is properly mixed, you have to cool it to 65 degrees centigrade and you pour it into a mold like this. So here you can find that we have placed combs over here. So what is this comb for? Because we need a hole to load the samples, isn't it? Otherwise, where will you load the sample? So that is why we place combs over here. So you allow it to cool. So once it is cooled, after some time you can remove this gel from here. So when you can see, where you can remove the comb, then you can see you will have holes over here. This is called as wells. So it is in this well only you will place your DNA sample or RNA sample or protein sample. So then after placing it, after loading your sample, you will keep it inside the electrophoresis unit. I will show you how the electrophoresis unit looks like. See, this is the electrophoresis unit. So you will take that slab, you will place it here inside the electrophoresis unit, and inside the unit only you will load the samples. You will not load it outside. So using the micro pipette, we used to see, this doesn't have the wells, so ignore that. So there will be wells in this agarose gel. So we have to load the samples over here and then we connect the positive and the negative to the power supply. So after some time, after loading the sample, what happens is, DNA being negatively charged, isn't it? We will load it at the end of the cathode. Which is the cathode? This is the cathode, right? So we will load it here. The wells will be here. We will load the samples over here. So after passing electricity, what happens is, DNA being negatively charged, it will move away from the cathode towards the anode. Okay, so it will start moving over here. So depending on the size, okay, so it is, agarose gel is nothing but a sieve. Okay, just consider it as a sieve. So what happens when you put something into a sieve? The lighter or the tiny particles will move fast and you get it first, isn't it? Whereas the heavy particles will take certain time to move. Similarly, here the DNA particles which are tiny in size, they will move faster and they will reach, move very faster. Whereas the heavy molecules will move very slowly. So that again we will remove it. We will place it on a, see, this is how, wait, before I go further, see, this is how, this is the electrophoresis unit, how it looks like. This is the agarose gel. Okay, so here you can find the wells. So in this wells we are loading the sample. And then after the movement, if you take that gel and you place it on a trans illuminator, you can see to what extent they have moved. So these are the wells. Got it? These are the wells. This they have done it in two places they have done. So you can find these are the wells. So depending upon the molecular weight, they would have moved. So higher the molecular weight, they would have moved less, whereas lower molecular weight, they will move very fast, isn't it? You can find even in running race, people who are very lightweight, very tiny, they can move very fast and for a farther distance, whereas people who are a little bit healthy and they cannot move, who are obese or something, they cannot move as fast as the people who are lightweight. So like that, here you can see they will move the DNA depending on the molecular weight and the charge, they will move, and we can remove that gel, we can place it on a transilluminator like this, and we can study the DNA. Okay, so this is the procedure how the agarose gel electrophoresis is done. So first the agarose gel is prepared. Okay, then it is poured into a tray which the comb which has a comb, right? You will have a tray, on that you will place a comb to make the wells. You pour it into that tray, then you will place it into a electrophoresis unit. You will pour buffer solution, then you will load your samples inside the well, and then you connect it to the electricity source. Okay, you run it at a constant voltage, and then finally we view the DNA on the UV light box, that is a transilluminator. We will place it on that, because otherwise through naked eyes you can only see some random, like not random, you can just you cannot see it very clearly. So you will place it on the UV light box and to observe the bands with much clarity. Fine. Okay. So we do Western blotting for proteins, isn't it? Western blotting is done for proteins, and Northern blotting is done for RNA, and Southern blotting is done for DNA. So what's the difference between agarose gel electrophoresis and blotting? See, in agarose gel electrophoresis, we stop at this step, right? We do all the procedure and finally we place it on the UV transilluminator, or here, just a minute. Or we place it on the UV light box, which we call it as UV transilluminator, and we study it. So with this, agarose gel electrophoresis is done. But in blotting, we go one step further. So we will take a nitrocellulose membrane. On this, we will place it so that the DNA molecules get transferred to that blotting paper, and we will use that for study. That is called as blotting. Okay, so even in blotting, we have all these steps. We have to prepare the agarose gel, we have to keep the combs in that, we have to pour it, we have to make the wells, we have to keep it inside the unit, fill it with buffer, we have to load the samples, everything is there. Okay, till here everything happens. Whereas in blotting, we have one more additional step. So we will take a nitrocellulose membrane, we will place it over this so that all this gets transferred to the nitrocellulose membrane, and we will further study that. Okay, so blotting is generally, in blotting what we do is the separated molecules are pressed against a membrane, okay, and study that. So Western blotting is done for proteins, Northern blotting is for RNA, and Southern blotting is for DNA. Why is it called Southern blotting? Can you tell me? Why is it called Southern blotting? Fine. Chemical methods, how exactly agarose gel electrophoresis or blotting, Northern blotting, Southern blotting is done. Do not concentrate. You know what exactly happens there, but do not try to break your head for technically how exactly it is done, because those are all in depth. It is not say some social issue which you can just speak, right? So it requires specialization to study all that in depth. So with whatever material that I have given, that is more than sufficient. In fact, that is more elaborate. You cannot find even so much material in PNAT or other physical anthropology books, right? I have given you very elaborate material. You can read that, and you can study what is different biochemical methods like paper chromatography, HPCL, agarose gel electrophoresis, blotting, and what are the various biochemical methods. Okay, so what is biochemicals? So what we are studying over there. So how DNA, RNA are studied. Simple, very simple terms you can learn it, and you won't be getting more questions also from this. So yes, I won't say completely there were no questions from this topic at all, but very, very rarely you will get questions from this topic. So don't worry much. Okay, All okay? Shall we proceed? Fine. Next, moving on to immunity. Moving